The cell cycle is a series of steps where the chromosome and other cell materials double to make two copies. The cell then divides into two daughter cells. The cell cycle is complete when each daughter cell is surrounded by its own outer membrane. Teach Me Sociology.com and Sylvia Matter from our book go over the four-stage process of the cell cycle. Interphase is the longest phase in this process and consists of 3 stages. During gap 1 or G1, the cell doubles in size and increases the number of organelles to prepare for DNA synthesis. During the synthesis stage or the S stage, DNA replication occurs. Before replication, each chromosome has one double helix DNA. After replication, each chromosome has 2 double helix DNA. Next, we enter the gap 2 or G2 phase. And during this phase, the cell continues to grow and synthesize the proteins needed to assist in cell division. We also need to include that the cells also can enter the G0 phase. The G0 phase are not preparing for cell division and can either remain in G0 or gain what it needs to proceed to G1. And if it cannot gain what it needs to enter to G1, apoptosis will occur. Now we enter mitosis stage, which has 5 phases. During the first phase, prophase, the condensation of chromatin occurs. Chromosomes are visible, but they are scattered everywhere. So they're just everywhere. And the nucleus disappears and the nuclear envelope fragments and the spindle begins to assemble as 2 chromosomes migrate away from one another. During the pro-metaphase, chromosomes are condensed but not fully aligned at the metaphase plate. They start to align like this. And kinetochores appear on each side of the chromosome, and these attach sister chromosomes to the kinetochore spindle fibers. And next, there's metaphase. During metaphase, the chromosomes are aligned at the metaphase plate. They're all in like a single file line. And at the M checkpoint, delay start of anaphase until each chromosome are attached properly to the spindle fibers. Next, we have anaphase. The chromosomes start to move towards the poles of the spindle, and the 2 sister chromatids of each duplicated chromosome separate to the centromere, giving the opportunity for 2 daughter chromosomes to move towards these opposite poles. And lastly, we have the 5th phase of mitosis. And each of the daughter cells are located and attached to the pole. The spindles disappear and new nuclear envelopes form around each daughter chromosome. And these contain the same number of chromosomes as the parent cells did. Then the division of cytoplasm requires autokinesis. And that is how the cell cycle and mitosis work.